Listo.
Okay. Good afternoon from London and good morning to Mexico. Welcome everyone to another session of the student seminar series from UNAM, UK. This is organized by the Center for Mexican Studies, UNAM, Reino Unido, in collaboration with the Mexican Society of Students in the UK. My name is Farid Bustos. I'm president of the Mexican Society of Students in the UK, and it is my pleasure to have you with us today. Today's talk is entitled From Neglect to Conservation, Genomic Insights into Chayote and its wild relatives, Sechium Cucurbitaceae. And today's speaker is Cécile Truchot. She's a final year PhD student in Doctorado en Ciencias Biomédicas or Biomedical Sciences PhD at UNAM. She is based at the Institute of Ecology. Her research is mainly focused on the evolutionary history, population genetics, and conservation of chayote and its wild relatives. This aims to generate knowledge to guide conservation strategies and contribute to food security. Cecile has also carried out research stays at the Royal Botanic Gardens, the Kew Millennium Seed Bank in the UK, and the Royal Botanic Garden in Sydney in Australia. She lived most of her life in France, where she earned her bachelor's and master's degree in at the University of Burgundy and Agro-Sud Dijon. Her academic trajectory combines international experience in both Mexico and France, also the UK and Australia, as I've mentioned, with a strong focus on plant genomics, conservation, and evolutionary biology. Well, as some of you may know, this is a 45-minute session for the presentation. Then will be followed by 15 minutes for questions and answers. And if you're joining us in Zoom, please write your questions in the Q&A or the chat box. If you're watching on YouTube, please send your questions through the comments, and we'll make sure to pass them along to our speaker. Cecile, it is an honor to have you with us here. Thank you for joining us today, and the floor is yours.
Thank you very much for the presentation. Also, thank you very much to Antonio Del Rio for the invitation and also the opportunity to share my work. So I'm going to share my screen. I hope it's working. Okay, so today I will present the result of my first chapter of my thesis. So it's more about genomics into chayote and the wild relative. So to start, I would like to bring you back to Mexico. And Mexico harbored between 10 and 20% of the world known species, and it represents only about 1.4% of the Earth's land surface. So even if it's a low surface, it harbors a lot of diversity. So Mexico is recognized as one of the megadiverse countries. So you have 17 countries in total recognized as a megadiverse country, and together they hold around 70% of the Earth's biodiversity. And they also hold endemic species. So endemic species are species that you can find exclusively in a specific geographic region and nowhere else on the Earth. So Mexico is one of the 8 main centers of origin and domestication for over 130 plant species. So 25 of these species are globally important for food and agriculture, such as corn, vanilla, cotton, avocado, and pumpkins. And you have also the chayote. So I know that chayote is not very popular in Europe. So if you don't know chayote, it's totally fine. But if you are coming from Mexico, I hope you know at least the chayote. So chayote, it's like watermelon, melon, pumpkin. It's from the Cucurbitaceae family, but it's classified in another tribe, and it's part of the genus called Sechium. So you have 100 to nearly 1,000 of underutilized species. So they are classified as neglected and underutilized species, or NUS. And most of them are unknown or ignored. So they attract little research and development attention. And you have to know that in reality, fewer than 200 species make a significant contribution to food production, and around 9 crops account for about 60% of the world total crop production, such as corn, rice, or wheat. And you may have heard about Sustainable Development Goals. So it exists around 17 Sustainable Development Goals established by the Food and Agriculture Organization of the United Nations. So they have a target horizon to 2030, and it covers topics ranging from ending poverty and hunger to protecting the planet and promoting peace and justice. I'm talking about this because the FAO make research called Rediscovering Hidden Treasure of Neglected and Underutilized Species for Zero Hunger in Asia. So it's the target number 2. And they identified the chayote as one of the 39 future smart food crops. So what does it mean? It means that they are resilient to climate change and harsh conditions. They also are nutrient rich, so they contribute to food and nutritional security. They also require very low fertilizer, water, or care, and can thrive in a degraded soil. And they are also very important for improving adaptive traits. So you have also, so the nurse can also contribute to other targets, such as no poverty. So most of the family have a chayote in the land, so in the backyard. So it's not only an agriculture, extensive agriculture they can have at home and make a source of income more the work that they have. They also can contribute to no hunger. So it reduces the dependence on a few global crops such as rice, wheat, and maize. Also good health and well-being, because nuts such as chayote are often rich in vitamins, minerals, and antioxidants. Also for the number 12, responsible consumption and production. So it also supports the cultural heritage and community resilience. Also for climate action, the number 13. So it enhances resilience of agricultural system to climate change and are tolerant to climate stress, as I said just before. And also the number 15, life on land, because it reduces the pressure on monoculture and preserve agricultural genetic diversity. I would also add that it helps for the number 5, so gender equality, because most of women also participate to the agriculture of chayote. So as I say, it's classified as neglected and underutilized, but it exists also other cucurbit species in the case of such as the chayote. So in the figure 2, you can see other cucurbit unknown. So I don't know also because I'm not studying them, but maybe you know them. And so it's valuable for the economic and ethnobotanic value. And yes, for the community and small town. Also, chayote had the highest production rate in Mexico for the last 14 years. So before the chia and the prickly pear. And of course, Mexico is the current world leading producer and exporter. and the main region states, it's Veracruz, so it represents 90% of the total production. And the taste. So most of people will say that chayote don't have taste. And I'm not agree for the spiny one, at least. So I feel like the chayote tastes the artichoke. But when I'm saying this to people, no one believe me. And I will count you a little anecdote. So I was reading a book about chayote. So you can look at the date. It's around 100 years ago. And I was not expecting this because I found this, that in Europe, it was used in London and Paris to the cuisine and other larger city in Europe to replace the true artichoke. So I'm not too crazy. And also the taste depend on the morphology because, for example, the white one that you can see, people report that they use it as a substitute of potatoes. So you have a lot of morphology and it exists around 10 varieties right now. And you can also find called land races, that morphology that don't fit into these categories. And I can highlight the work of Marianne Angeles Cruz. She's an undergraduate student and she's also currently studying the morphology of these land races and also the wild chayotes. So where does the chayote was first domesticated? So we don't have any archaeological remains because the seed is very soft and decompose, and so we don't have any proof that the chayote was here in Mexico before, archaeological proof. But we have in a codex some painting of chayote. So I don't know if you can see here in the screen. So here you can see the chayote here and here. And it was described in the Mixtec culture and also the Aztec culture. So here are located the Mixtec and Aztec culture in Mexico. And also it exists a study about linguistic. So the word chayote means spiny squash, and it's from the Nahuatl. So the Nahuatl is the language of the Aztec. And also they made a linguistic study, and this linguistic evidence suggests that the chayote could be first cultivated by the Zapotec speakers between 1200 and 500 before Common Era. So here in the cycle. So it exists a lot of civilization in Mesoamerica. So here is a timeline when you can see the Mixtecs, the Zapotec, and the Aztec that I said. And if I talk about domestication, I can also talk about a wild ancestor, because domestication, it's an evolutionary process driven by human selection pressure on wild species for human need. So when you think human need, you maybe not think about the little cat or a cute cat or a flower that was domesticated just because they are beautiful. And if you want to know more about domestication, you can read this paper. It's well explained. But I'm back to the corn, and I don't know if you know the wild ancestor of the corn, but it was looking like that. So it's called Teosinte. So this is an example called, it was at the Kew Garden in the herbarium. So the species, the specimen was here since a long time, but it's well preserved. And also, I will talk about domestication syndrome. So it's a convergent change that you can see in all domesticated species. So most of her domesticated species lost their natural dispersion mechanism. They also have an increase in size of the edible organs, such as grain, fruit, and tubers. Reduction also of seed dormancy and reduction of natural difference, like the spine, and also morphological change. So the shape and also the color is getting more attractive. So again, for the example of the corn, I can show you all the diversity and the colors and shape for the corn that you can find only, well, I think only in Mexico, because in Europe, I never seen the red one, for example. So like the chayote, like the corn, a chayote also, yes, have a wild ancestor. So I would like you to imagine a wild chayote, if it have a spine or not, which color it should have, because chayote go to dark green to white. So these are the wild species. So if I take again the list of the characteristic, you can see also the increase of the size of the edible organ. So for example, the fruit. Also, the chayote, you can eat everything. You can also eat the roots, the entire plants. You have also in chayote a reduction of seed dormancy and a reduction of natural defense, like cucurbitaceae. It's the molecule that make the fruit very strong, and you can't eat it. But about natural defense, about, for example, the spine, chayote have some varieties or land races with spine and other without. So it don't have a totally lose the spine. And morphological change, because the color, you don't have a white wild species, and also you have one very long. So this one you can't find it in the wild species. So here are one by one the wild species. So Sechium hintoni, found in Jalisco. Sechium compositum in Chiapas. As you can see, the square are also the place where we went to collect and the species. Sechium chinantlense in Oaxaca. And the subspecies, the wild subspecies, called Sechium edule subspecies silvestre. You can find it in Veracruz, Oaxaca, and other state, Puebla. So why it's so important to resolve phylogenetic relationship between cultivated and wild species? It's because you can know more about evolutionary biology, and also it serves a lot for genetic improvement. So for the chayote, which one of these are the wild closest wild relative? A lot of study try to resolve this. So we had a morphological study and also with molecular markers, and the number of species of the genus also vary a lot. So to 3 species to 11 species, and they even removed the genus. So I will start with morphology. So with morphology, the closest wild relative was estimated to be the Sechium edule subspecies silvestre. So that's why also they called it subspecies. And also Sechium chinantlense was also very close, but because it have a distinct organization in the male flower, in the filament morphology, they classified it as a distinct species. And then come the era of molecular markers. So you can think that molecular markers will help to to know and understand well the relationship between them. And finally, it was the contrary, because it removed everything, because they could find the wild species with the cultivated one together. So it just make more questions. And also they could find hybridization between two species. So one of the theory or one of the purpose or also proposition was to fusion these species. So all of these species should maybe one species. So that's why I was focused on this area on the first chapter. It was to know more about the relationships between the cultivated and the wild species, and particularly how many species comprise the Sechium group, and also if they are the same species, as I said before, or not, to understand the phylogenetic relationship within the group, so to know which wild taxon is the closest wild relative to Chayote, and also to estimate the divergence time for the crown node of the Sechium clade. So to collect, we used Encyclopedia and global biodiversity information facilities to help to identify the collection sites. And it's not working. I don't know if it's my connection. Can you hear me?
Yes, we can hear you.
Okay,
Yes,
because
yes,
it just stopped to it's not moving. I'm sorry.
Don't worry, I can see the mouse moving.
Yes, but it's completely blocked. I will stop to share and start again to share. Yes, okay. Can you see my screen or not?
Not yet.
Okay. And right now?
It's charging. I can see your presentation.
Yes, it's work again. I don't know what happened.
Okay.
So yes, we use Encyclopedia and Global Biodiversity Information Facility to identify the collection sites. So here are the location of each of the taxa. So in green, you have the Chayote, so the cultivated species. And also we include the contact zone. So to know if we can have a signal of hybridization between, for example, Sechium hintoni and the cultivated one, the Chayote, here also, and also in Chiapas. So we use then technique called genotype by sequencing. So it's a reduced representation of the genome. So here you can see the DNA, and we use enzyme to fragment the DNA. And then different platform can be used, but we use Illumina sequencing. And the final number of loci depend on the dataset. So it depends on the number of the individuals, the type of analysis, and also the filter applied. And this technique is very useful to estimate genetic diversity, population structure, and phylogenetic relationships at recent time scale. So here are the results of PCA. So PCA shows the percentage of the total genetic variance. So the question was if the species were one or different, still different. So here you can see an example of one species but different location. So the individual are colored by the population, but you can see that they are together, clustered together. That means that it's one species. So as you can see, our results show that each group are genetically distinct, because otherwise you should see, for example, the pink point with the blue and green, and also this one, orange and brown, all together, all clustered. And this is not the case. Then we did a structure analysis. So the structure analysis show the ancestry proportion given a chosen number of cluster. So for example, you have a 4 cluster here shows. So you ask the program to assign the ancestry proportion for an individual. So you can see cluster 4, 5, 6, 7, because it's also good to explore the different car number. And also again, as you can see, all are well defined. For example, here you can see in the Chayote that one individual have proportion ancestry of one location of the wild species, but it don't look like the wild species, the subspecies Silvestre. So again, we don't find clustering that all the species are one. And now I will talk about FST. So the fixation index is a measure of population differentiation due to genetic structure. So here I will put the different location of the population. And an example, here it's the population of Chayote. So it's the same species, and you can see that the value are very low. So 0, it's no differentiation, and 1, it's complete differentiation. So here it's the same species, so it's normal because it's different location of the Chayote. Here, between the two population of Sechium compositum in Chiapas, you can see a value of 0.25, so it's still okay. Also, I mean, it's still okay because a rule, it's not a rule, but when it's an indicator. So if you have more than 0.3, you can consider them as a distinct species. So here you have a value below 0.3, so it's the same species. And then if you compare, for example, Sechium compositum and Sechium chinantlense, you have a value bigger. So the idea, again, to cluster Sechium compositum with Sechium chinantlense, the subspecies, the wild subspecies, and the cultivated one, here it's not well supported, so they are genetically distinct. And then we also did another analysis, so it's a hypothesis. So the idea to cluster all these species are the number 6, and it also get the lowest value of support. So which wild taxon is the closest relative to Chayote? Here you can see that it's the subspecies, the wild subspecies, followed by Sechium chinantlense, and these 3 species have an apical slit. So you can see here the apical slit in the picture, and it's the only one on the genus to have them. And you can also see alternative topology, so it can be incompletely enlarged sorting, that it happens when you have a short time between speciation. And maybe this could explain the result found before, because it was a different type of markers, and they couldn't have the resolution of this tree. And also we estimate the clade again. So we used data from the gene bank, and we found that the age was a bit younger. So the species of the speciation of Sechium may occur during the Pleistocene. So it's a period characterized by repeated glacial cycles to expansion contraction that fragment the cloud forest. So it's the habitat of the wild Sechium species, leading in some case to isolation and also speciation, such as Sechium species. So if I go back to the first question, if they are the same species, our results don't support the idea of fusing the different species into one. And also it can highlight one question: if a trace of hybridization where they are not observed under natural conditions are really sufficient to justify to remove the species status for certain taxa. So here are examples of species that have a distinct distribution range and show clear morphological difference. And they have a hybridization between species of the same genus in the natural habitat, and also still keep the status of species. And removing species status also it's important for conservation. So now I will talk about also conservation, and I will start to talk about climate change. So climate change and its impact, it's one of the most critical challenges for biodiversity and conservation. And you may have heard about the Paris Agreement. So it's an agreement that was signed during the COP21 in 2016. And the aim was to keep the global warming below 1.5 degrees compared to the pre-industrial level. So pre-industrial level are in the era of 1850, 1900, yeah. And why 1.5? Because 1.5 degree was a critical limit beyond which the economic and social and ecological consequence of climate change could be increasingly severe. So one question for you: do you think we already exceed the 1.5 limit, or are we still okay? So maybe you know, maybe you don't know, but unfortunately, we passed this threshold last year. So 2024 was the first year to pass the 1.5 global warming limit. And here you can see the curves of the rising temperature during the years. And this threshold was initially projected to reach around 2025. So we reached it last year, so 1 year before. But also this prediction was the most pessimist scenario in 1999. So we can say, well, it's very scary. I don't know how to say. And it's also good to know this because you can make prediction about the future distribution range. So here it's a potential distribution range, and with the prediction, you can also make the future distribution range. And this was made in 2009, and the prediction for 2060 is that Sechium chin and Lindsay will not survive because it don't have any remaining area. And also it's very critical for Sechium hintoni. Sechium hintoni was a species declared as extant and then rediscovered. So it's also very critical for this species. And when we went to collect, in some cases and some places, we couldn't find at site when it was historically recorded. So it can be because, as we saw before, the reduction of the area distribution, but also some sites were converted into residential and agriculture area. And so to conserve and to preserve the species, of course, we can't do it by in one day. It can't be done in one day, but we can elaborate a short-term and long-term strategy. So one of the short-term strategy is to evaluate genetic diversity. So genetic diversity is very important because it enables species to adapt environmental change, resist disease, and avoid problems associated with inbreeding. So inbreeding is known for example, the kings. So because it reduces survival, fertility, and also can lead to malformation and disease. And also it helps to inform conservation priority and ensure the long-term survival of population to evaluate genetic diversity. So evaluating it's very important. And also we analyze different value and measurement. So one of them is the inbreeding coefficient. So as you can see in the table, you have a value below 0. So below 0, it's a strong outbreeding and mixing of highly divergent population. 0, it's parmixia, so it's random mating. And 1, it's the maximum inbreeding. So maximum inbreeding, you can find it in the Chayote. So it's 0.3, so it's not 1, but it's still positive. And it's common to find higher value in domesticated species because humans select and propagate only a few individuals. So it reduces genetic mixing and increase inbreeding. So it's quite normal to have this result. And these results are coming from expected heterozygosity and observed heterozygosity. So the expected one, it's the theoretical genetic diversity in a population if individuals were mating randomly. And the observed, it's the actually observed in the population. So here are the formula. And if you want to know more about formula and population genetics, you can also check on this canal because we have a lot of classes of genetic population, and also they explain all this measurement. So if you have a look of the value of the genetic diversity, you can see that the lowest value is for Sechium hintoni, so the species that was declared as extant. So it's also very preoccupant. And we have also another estimation, so the estimation of effective population size. So first, you have the census size, so it's the total number of individuals in a population. And then you have the effective, the effective size. So it's the number of individuals in an idealized population that shows the same level of genetic drift or inbreeding as a real population. So it's like the genetic contribution of individuals to the next generation. So how you can estimate it, or with genetic data or without. So you have a ratio. So the effective population size can be approximated as a ratio of the census size. So it's around 0.10 for most of the species, but it can vary. So we estimate the effective population size, and effective population size is one of the key indicators of the coming memorial framework. So it's to stop and reverse biodiversity loss by 2030 with a long-term vision to 2050. And this reflects the ability of population to maintain its genetic diversity and resilience to environmental change. So the value N50, it's important because it represents the minimum recommended for population size for a short-term survival. So it ensures the maintenance of sufficient genetic diversity to face disease and climate variation. So we estimate it, and it's below 50. So it's also very critical. And we have the long term, so we don't, so we already don't pass the 50 threshold, but ideally it's to be up to 500 to ensure persistence over time. And to generate this data, it's very important because it can allow the inclusion of the red list or the equivalent of Mexico. And it's a step toward their protection because you have a different level, and with the data you can choose, you can put the species in this different category. So for Sechium hintoni and the other Sechium species, it should be at least in the threatened categories. But this depends also of other data, and yes, people choose. And this is for the Mexican one, so the NOM 059 Semarnat. And for the long-term strategy, so you can think about to conserve it in the seed banks, such as the Millennium Seed Bank at Kew. And you have different categories. You have the orthodox one, so it's when you don't have too many complications to conserve them. Then you have the category of intermediate and recalcitrant seeds. And Chayote are part of this species, recalcitrant seeds, because the seeds are damaged when you removed it and don't have a lignified seed coat. So one of the action or proportion, it's to have an in vitro collection to ensure the conservation of Chayote and the wild relative. Also in vivo, you have the germplasm bank. So in Mexico, you have the Banco Nacional de Germoplasma de Chayote, located in Huatusco, Mexico. And also you have this paper about cryopreservation of in vitro shoot tips of Chayote. So you have the research, you have research on the conservation of Chayote. And long-term strategy, so has a new species, it's to do educational workshop and also divulgation to make public known about the importance of preserving wild species. Also to encourage the adoption of less invasive farming techniques that minimize environmental impact and support biodiversity. Also to collaborate with the authority and local community to protect the area and make a natural reserve to conserve the habitat and species. Also, the inclusion in botanical garden to avoid any risk of introduction of other Sechium species, and also avoid hybridization. So yeah, a botanical garden is also one of the proposal. And also reintroduction program, so to restore population in historical habitat where the species has disappeared. And so the acknowledgement, so I will thank the Institute of Ecology the FEST Istacala, Conabio, and Scientia y Tecnología, so CCT, for the funding of this research. And also my main supervisor and my committee, so the Dr. Luis Eguiarte, Rafael Lira, Juan Pablo Jaramillo, and also Alicia Mastreta, that she was my Q-internship supervisor. And also people that will contribute and will contribute to this analysis and data. And I hope that the paper will be published soon. And also big thanks to the Laboratory of Evolution Molecular y Experimental of the Institute of Ecology, UNAM, because also they helped me to collect and also for the good vibe. And thank you for your attention.
Thank you. Thank you very much, Cecile, for this very interesting and fascinating talk. Now we'll open the floor to questions from our audience. Yeah, so if you're watching this in YouTube, just write the question in the comments, and then we'll pass that to Cecile. As of now, I want to ask you something. So you did your bachelor's in France, the master's. Obviously, there's not chayotes there, but were there any challenges you faced given this situation? Uh, the seeds were perishable. What was there anything that happened during your research?
So yes, the fact that the seeds are perishable was also it complicates, for example, for DNA extraction or to conserve the species, because we had when we went to collect, we had to make the DNA extraction very quickly, or we had also another alternative. It was to put the samples in the freezer, but we had also to make the photo, to take the picture, to also make the measurement. So it's much more easier to have, for example, seeds to conserve than perishable seeds. And also, maybe, yeah, for conservation, it's a challenge. Oh.
Yeah, unmuted, sorry. This question comes from YouTube. It says, in your view, what policies or initiatives would be the most effective to protect these neglected and underutilized species in Mexico?
I think a lot of divulgation, popularization, because finally, I think we can generate that data, but the most important thing is to talk about. And yes, just to make people know about these species, that they are important, and in Mexico, but also overseas, because nurses are everywhere, finally.
So I'm trying to, well, it's probably not the same, but it could connect in some way. Uh, there should be some initiatives and policies for protecting these neglected species, but is there anything, or does your work connect for some other efforts for crop conservation, diversity, strengthen these food systems, or anything related to these, probably helping poor communities food-wise?
Sorry, so the question is to know
If your work connects with some other efforts that can conserve crop diversity and or strengthen some
uh,
food systems.
we will, so we are already working on, it's about the land races. because also land races have a lot of genetic diversity, but first that's why also Marianne, that I show you the work, it's start to make morphological studies, and then should be also genetic studies, genetic diversity, to know more about the land races, and also to highlight these land races, because using only varieties don't help to this support of the news.
Okay. Thank you. So, well, and um, this could be a little bit more for the audience that is just finishing their bachelor's or master's, but so there's quite a huge spike on these biomedical, biotechnological, engineering degrees, on scientific degrees overall. What advice could you give them if they are interested in working with these underutilized crops?
Sorry, can you repeat advice to?
Yeah, what advice could you give to these young researchers, bachelor students that are interested, or that they want to work with neglected or these underutilized crops?
The advice, it's maybe to try to make the news, to highlight them, because I know that it's not very easy to work on unpopular crop species, because, for example, when I was in a congress, or when I'm, for example, in other talking to people that are not coming from Mexico, it's why are you working with this species? Or so I think it's to keep studying these species, think about all the valuable data, and also for people, because all these data will be very, very valuable for the people that are living with these species, that have a source income for these species. So yes, it's important to generate this data and to protect them and to promote them.
Okay, thank you. And I think you might have partially responded. The next question it's how did these international experiences you've got, bachelor's and master's in France, this internship in the UK, also in Australia, how did this shaped or shaped your approach to this research topic, the Mexican biodiversity?
So it was very rich in experience, to speak English and Spanish. So maybe you are afraid to go there, but finally I maybe should recommend to any student to go overseas, even if you think you can be alone or lost because of the language. But if you try, you will have friends, and you will have life. So just keep going and go, and it's very a beautiful experience.
Thank you. And finally, or this would be my last question, I think. Uh, do you think that is there any potential with the Chayote for tackling food insecurity and promoting some sustainable agriculture in Mexico?
Yes, because it's already it have the rate of production of chayote grow the last years. It's also very exported overseas, United States and also Europe, because when I was in London, I could find chayote. So I know that have a potential also in India, in some part of Asia. Also in Asia, they were making studies about the compound of chayote to reduce lipids. So for medicine, also the Y species, I didn't talk about here too much, but some research about cell cancer to fight the cancer in some cells. So, and when I was looking also about the paper about chayote, I could see that a lot of work were published before the year 2000. Then it started to don't have a lot of study, and if you have a look to the maybe 4 last years, the publication of chayote start again. So, and in India, in Japan. So yes, chayote, I hope that it will continue and will still attract people.
Okay, this is yeah. Hopefully there's gonna be more publications on chayote and some other Mexican crops, Latin American crops. And I think too, it's quite important to continue to do some research on this, especially if they could tackle food insecurity or the world hunger overall. Uh, I'm a bit curious. What were some of the most surprising findings that you found during your research about these chayotes and their relatives?
Yeah.
Yeah. Let me rephrase that probably. What were some of the most surprising findings you found during your research about chayote
Sorry, I
relatives? Yeah,
didn't
no, don't
hear
worry.
the question. The I was when I start the analysis, because as you see, they had a proof of hybridization. So I was like, maybe I will find more proof of hybridization, and finally, not too much. So this was the surprise, that maybe I was expecting more proof of hybridization, and finally not, because it's understandable, because we went to the natural habitat. So you can have hybridization, but after you have stepped, no? So maybe it can have a hybrid, but can't survive in the natural habitat. So because when they could make hybridization, it was in a center, and it was the purpose to make hybridization. But in the natural habitat, maybe it can happen, but they will not survive, or maybe they can't reproduce with the other one, and can't participate to the genetic. Then you don't have any trace of it, because it don't reproduce. So this was maybe the most surprise, that I was expecting maybe more proof of hybridization on the natural habitat. We can't hear you.
Yeah, was muted. Sorry. I think that's quite interesting. Thank you. Thank you very much for this very interesting talk. Uh, congrats. It's a very
Thank
interesting
you.
and quite a complete explanation, and I'm conscious of time. On behalf of the Centro de Estudios Mexicanos, UNAM UK, and the Mexican Society of Students in the UK, we thank you, Cecile, for this excellent and very interesting presentation, and all of you for joining us today. We invite you to follow the upcoming activities through UNAM UK social media. You can find them on X, Blue Sky, Facebook, Instagram, and LinkedIn as UNAM UK. And thank you very much, and see you in our next seminar.
Thank you so much. Bye.